morning, respected Professor Murlidhar sir and the dear participants, teacher friends. We are on fourth day of refresher course supported by Science Academies, and Academies have proudly announced that Department of Science and Technology, Government of India is celebrating the 75th year of independence, and on the eve of 75th Independence celebrations. Refresher course is also one of the part of celebration, and it is called as Azadi Ka Amrit Mahotsav. So, with this words, I would like to introduce our speaker, Professor K. Murlidhar sir. He is a distinguished biologist of our country and was born at Coimbatore, Tamil Nadu. He obtained his BSc and MSc from Usman University, Hyderabad. He started his research work under K.L. Ramchandra sir at Usmania University in summer of 1969. He obtained his PhD from Indian Institute of Science, Bengaluru in 1976, working under N.R. Mudgal. His PhD work demonstrating that beta subunit of luteinizing hormone can bind ovarian receptors fetched him the Professor K.V. Giri Memorial Gold Medal for the best PhD thesis of the year. He was selected for the first leadership lecturership, first lecturership in the School of Life Sciences at newly founded Central University of Hyderabad in August 1976. During 1979 and 81, he was research associate in the Department of Cell and Molecular Biology at SUNY Buffalo, New York, USA, with O.P. Bhal. His work led to the development of most sensitive radioimmunoassay for HCG, a pregnancy hormone. He also demonstrated the immunocontraceptive vaccine potential of DS5 HCG beta subunit. He joined the University of Delhi in 1983 as reader in biochemistry and became professor in endocrinology in 1988. He was chairman of the department during 2001 and 4. Sir has initiated long-range research program on water buffalo endocrinology. His laboratory has developed procedures for indigenous production of buffalo hormones like LH, FSH, TSH, GH, and PRL. Sensitive homologous immunoassays and heterologous bioassays for these hormones, besides, besides characterizing their physicochemical and immunochemical features. The laboratory was among the first to report LH and PRL were sulfated, and that this contributes to microheterogeneity. His laboratory later discovered the presence of tyrosine O sulfate in sheep buffalo PRL. Using biophysical techniques, his group has studied the protein unfolding and buffalo, folding of buffalo GH, naturally occurring size isoforms of buffalo prolactin, cathepsin derived peptides from buffalo prolactin, and a synthetic peptide based on the internal sequence of buffalo prolactin were discovered to be powerful inhibitors of angiogenesis stimulated by VEGF or bradykinin. His group was published over 150 articles in peer-reviewed journals and books as chapters. Sir is an outstanding teacher and has taught courses in biochemistry, gamete biology, immunology, endocrinology, cell biology, and medicinal chemistry at University of Hyderabad, South Asian University, and at University of Delhi. So far, he has mentored 27 PhD and 27 MPhil research students. He has published over 150 articles in peer-reviewed international and national journals and edited numerous books. Sir was chairman of the UGC Curriculum Development Committee for Zoology and brought out a benchmark report on MSc and BSc in zoological sciences. For NCRT, he brought out the new textbooks for higher secondary school biology in a completely new format backed by a sound philosophy. Amazon, after proper survey and referring in 2017, ranked these books in the top 5 of 100, 750 such books in the world. He assisted University of Delhi in restructuring undergraduate science education and introduced a new honors course in integrated biology, which is first of its kind in the country. He was the convener of the NET exam and brought more balance in the examination, giving representation to all important areas of biology. He was the chairman of the PAC Annual Science of DST for 3 terms and nurtured research in this area. He was assisted all the funding agencies like CSIR, UGC, ICMR, DBT, DST, Ministry of Environment and Forest, Indian Council for Agriculture Research in various capacities. He has served on the research advisory committees of CCMB, IGIB, MRC, NBRC, AI, NII, besides assisting in the selection and promotion of faculty in a new number of educational and research institutions of our country. He was educated on more than 356 PhD theses from over 35 universities and research institutions. Sir is an elected fellow of all the science academies of our country, that is Nasi, Prayagraj, INSA, New Delhi, IASC, Bangalore. He has delivered the Sadak, Sadak Sharaswamy Endowment Lecture for SBC 1996, U.S. Srivastava Memorial Lecture for Nasi, V. Gopalakrishna Rao Endowment Lecture for Usmania University, Y. Subbarao Memorial Lecture for GGS Indraprasth University, Professor M.R.N. Prasad Memorial Lecture for INSA, Hargobind Khurana Memorial Lecture for GGSIP University, G.P. Sharma Memorial Lecture for Punjab University, P. Govindarajulu Endowment Lecture for Madras University, Platinum Jubilee Lecture for Indian Science Congress, and many more. Sir has served as a member of the Executive Council of NASI and INSA. He was the chief editor of the Proceedings of Indian National Science Academy for over 6 years when he transformed the journal and brought international recognition. He was chairman of the INSA National Committee for Cooperation with IUBS for a term. He was a Sir J.C. Bose National Fellow during 2014 to 16 at South Asian University, New Delhi. He was Jawaharlal Nehru Chair Professor during 2017-18 at University of Hyderabad. He was a member of SIR. He is presently on the advisory board of the Inter-University Center for Teacher Education at Banaras Hindu University and a visiting professor to Annamalai, Madras and Hyderabad universities. With this introduction of Sir, I would like to request Sir to start his presentation. Over to Sir. Good morning, Sir, and welcome on behalf of Department of Botany, Telangana University, and the participants for 14 days refresher course. Over to Sir.
Thank you, Dr. Khan. Am I audible?
Yes, sir, you're audible.
Okay. I will start sharing the slides first, and then I will start the lecture. Are the slides visible?
Yeah, they are visible.
I'll make it full screen.
Yeah.
Is this okay?
Yes.
Am I audible clearly?
Yes, sir, you're audible.
Okay. Thank you, Dr. Khan. And not just as a formality, at the beginning, I would like to thank the authorities of Telangana University, especially Dr. Halim Khan, and the director of this course, Professor Manoharachari, for thinking that I can also give a lecture in this refresher course. As I mentioned on the first day, I was hesitant to speak in this forum because I don't do any research in so-called systems biology or the phase 3 in the history of biology, what is popularly called as web of life and what it means. But for some reason or other, anyway, I agreed to deliver some lecture on this, and I will try to initially tell you a little bit about the history of biology and biology teaching and how this has created problems in our understanding of biology and how systems biology will come to our rescue. And I will take example from endocrinology where we can address this from reductionist point of view, from classical descriptive biology point of view, as well as from systems biology point of view, and see whether we understand better than earlier days. So that's what I wish to do it. And let me begin with, see, biology, all of you are aware, and I am sure you also tell everybody that it is a science of living organisms. It has developed enormously if you go through histories since the days of Aristotle. But although it is part of natural science, which is very clear with regard to methodology, with regard to what kind of questions you can raise, biology, for some reason, I will not go into detail. Yesterday, Professor Shivanna just hinted at that point, and for some reason, therefore, biology people did not follow the general principles of natural science, and they did not do a single experiment in their subject. We will not go into detail about all these things. Even the semantics of this subject, that means the technical words that they use, neither the teachers nor the textbook writers clarified many of these things. And therefore, basically, biology was being taught not as another natural science trying to understand the living process, especially in the so-called animal world, plant world, and microbial world. It simply became a sort of mechanism for transfer of information. Wait a second. And therefore, for a long years, even till today, biology teaching means information transfer. Now this, therefore, exchange of second-hand or first-hand information does not allow you to understand biology. This is the first point that I wish to make in the next few slides. And then we will come to how the new approach, systems biology, allows us to understand. Let us begin with, I have shown you some newspaper cutting where there is a patient who is lying in ICU and he is living on the so-called machines, heart-lung machine, artificial. His family is surrounding him, but he cannot recognize this family. And therefore, the doctors say that he is clinically dead because he is a brain dead patient. He is surviving, only physiological processes are being done by external machines. But the family, for sentimental reason as well as for philosophical reasons, does not believe that the patient is dead, that he is alive, and therefore they continue to look at him or her and then believe that it's a life. In other words, we don't seem to understand what is living and what is life. The belief in this particular case that I, as an example, is that life is a continuous process. Death is only an intermediate stage, that life is continuous, there is no discontinuity, and you enter another body and therefore continue the life. So an individual is recognized by his self-consciousness, and that is the way they look at life and living. And therefore, consciousness becomes a property or phenotypic marker by which you say that this is living. But unfortunately, this is a semantic confusion. What they are referring to is self-consciousness and not consciousness as such. Every living organism is aware of the environment. That is one of the fundamental definitions of living. And therefore, you see, we begin biology by promoting confusion about, because we don't understand what exactly is living. So let us try to understand why is this so much confusion of what is a living organism. Now, philosophy and science seek answer to the same question: what is the truth about this universe? As most of you are aware, in the very, very early days, all knowledge was called as philosophy, and whatever they know, man knows, he used to express. It's only later days that the philosophy or knowledge about this material world became separated, and that became science. In 15th century in Europe, during the Renaissance movement, this science was more standardized and it was called as natural science. There was a scientific method which was described. Many people have written about all this. I will not go into detail. All that I will say that people like Francis Bacon, René Descartes, Immanuel Kant, Karl Popper, these are all philosophers of science, and they have written extensively about what exactly is science and how to do this science. Let us understand two principles about it, two concepts. Science deals with the structure and function of nature. Nature means initially the material world, subsequently it included living organisms. This is a historical reason because scientists are also human beings and they believed that all living organisms are created by God, and we don't investigate living organisms because it's a God's creation. There is nothing to ask, nothing to understand, nothing to describe. For that reason, therefore, they restricted initially to material world. Later on, of course, the biosphere was also added. And therefore, this science about nature pursued through scientific method, that entire information as well as the way it is acquired was called as natural science. And physics, chemistry, biology are the three pillars on which it stands. Later, earth science or geology was added on that. But even today, many serious philosophers of science believe that biology and geology are not axiomatic sciences like physics and chemistry. They don't have independent existence. They don't have any independent set of concepts. All this has changed now, but I'm only tracing the history. So therefore, this science was essentially physics and chemistry from the 15th century. This is called modern science. This is also called Western science because this science originated in Europe, in Italy, England, France, Spain, Portugal, and other places. And therefore, this is called as Western science, as Europe is in our West. Physics, the first pillar of science, natural science, constructs the physical reality of this universe, the truth about this universe, through studies on the behavior of nature, and therefore what is the structure of this nature and how does it function. In this behavior, is there any pattern? And physics people discovered these patterns in the form of laws of universe. They also call it as universal laws of universe. This is the behavior of nature in a particular way. So if you open a physics textbook, Their philosophy and their discovery of the truth of nature is given as chapters: laws of thermodynamics, laws of mechanics, laws of optics, laws of electricity, and so on and so forth. And that physics is a philosophy which discovered the truth about the physical reality of this world, that this material world is real. But they did not investigate or do anything on biosphere, living organisms. As I said, the reasons are not very clear, but subsequently, when they applied physical principles and concepts to living processes, they realized to their remarkable understanding that living organisms are energy machines. Let me briefly tell you one of the greatest concepts in physics. These are the laws of thermodynamics, which says that every object in this world always moves from a non-equilibrium state to a equilibrium state. This is a fundamental principle, and the universe is subject to entropic doom. And equilibrium means change in free energy is zero. Therefore, a system at equilibrium cannot perform work. Living organisms perform work always till death. Therefore, death is equilibrium, and living is a non-equilibrium called steady state, because we have a lot of free energy and we work continuously. And therefore, this is the way physics people defined living, as a condition or a state where it can perform work. Till today, nobody has seen dead bodies doing voluntary work. And that's the reason this physics definition of living is absolutely correct. However, chemistry people, which is also another philosophy of science, asked different type of questions, and they wanted to know whether there is a universal substance, single substance in this universe from which all other objects and substances are derived, and therefore you see diversity of objects. They could not discover this universal substance. Ultimately, Mendeleev told that there are only 72 truths or 72 elements. So element is a truth for chemistry, and 70, today it is about 140 and odd. That should not confuse us. Basically, therefore, chemistry seeks truth about this universe in its composition and believes that an element is the ultimate truth, because copper is copper is copper, gold is gold is gold. One cannot turn one into another. Of course, all this has changed later because of nuclear physics and other developments. And this chemistry believes, when they applied chemistry to living organisms, they believed that these living organisms have chemicals, and that these chemicals, we call them biomolecules, they are continuously in a flux. This is called metabolism. And these reactions are of two types: anabolism, where a complex structure is biosynthesized from a simple compound, and catabolism, where a complex structure gets degraded. Physiologists call it digestion into simpler molecules. And as physics says, any degradation of structural complexity results in the release of energy. And therefore, chemistry people believed that living organisms produce energy by degrading food items, which are basically macromolecules, in the process of digestion and phase 2, phase 3 metabolism, and generate energy, store it, and use it to remain themselves in steady state. Therefore, metabolism becomes the defining property of living state, and they also confirm that living systems are energy machines. That is how a physicist or a chemist looks at living. Science is all about measurement. Unfortunately, biologists did not measure anything for about 1700 years, till the middle of the 17th century, when a philosopher and a mathematician called René Descartes told people that you have to do experiments, otherwise you will not understand biology. Then what were we teaching till then? It was descriptive biology. It was called phase 1 biology. Now it is called natural history. It is purely description of whatever you observe, either through naked eyes or through microscope or by any other means. And therefore, it's only after 17th century when people exhorted biologists that you should also ask questions and try to answer them by investigation. That means do some experiment and therefore measure something, that reductionist biology started, experimental biology started. Only in the late 18th century onwards, biologists started doing experiments seriously. And therefore, this is the phase two of biology or reductionist biology. Today, you call it as biochemistry and biophysics, molecular physiology, or molecular biology. And therefore, the people who were doing this kind of biology separated in the university as separate departments. The very first confusion was because biology was taken as simply a mass of information and teaching is only information transfer. Because the information was too much, biology was divided as botany, zoology, and microbiology. And that is the first confusion that was started. And the casualty was understanding biology. In the second confusion, the reductionist biologists, biochemistry, biophysics, physiology, genetics, and other areas, they separated from the parent departments because they thought they are different. And therefore, for a long time, reductionist biology in the form of biochemistry, genetics, and all were being taught independently from the classical biology, which includes taxonomy, systematics, and evolution. Incidentally, phase one biology or natural history, the biggest boost for that came when Darwin told us that the entire diversity that we see, biodiversity, is not constant. It has come from a single ancestral organism, and this process is called evolution. And this, he wrote a book in 1859, published it, got all the credit. Others also did, people like Wallace and others. We will not bother about details. So this Darwinian theory of organic evolution by natural selection is the biggest human concept in human knowledge, and it is the biggest concept in phase one biology. Reductionist biologists did not bother about it, and they paid the price for that, because any amount of biochemistry, any amount of physiology or genetics or anything will not allow you to understand. It will also be a description, only thing it is at molecular level. The reason why both these biologists, teachers and students missed understanding was that biology is characterized by certain properties, living organisms, and one of them is emergent phenomena. There are emergent properties exhibited by living organisms at every level of structural organization. In other words, if you look at living organisms and look at their structural organization, all of you know that we can have ecosystems, we can have populations, communities, we can have individual organisms, complex, we can have organ systems, organs, we can have tissues and cells, subcellular organelles, macromolecular assemblies, biomolecules, small molecules, like that. The entire living organisms, spectrum of them are organized in different groups at different levels of organization. And this essentially, the emergent phenomena in biology tells us that what you see at one level of organization is not seen in the parts which contribute to that at the lower level. That means what a cell does, no subcellular organelle does. Take secretion as an example. What a population does, an individual does not do. What an individual organism does, individual tissues and organs don't do. Therefore, what you see at a higher level is called emergent phenomena. The best example is mind is an emergent property or exhibition phenomena of brain. If you cut the brain and look at a torchlight, you don't see mind there. Mind is something extraneous to brain. This is one of the philosophical problems in biology, but no biology teacher teaches biology by questions and answers and raising philosophical problems. They take it as information transfer, and that is the reason we are in a bad state. Now, to understand this emergent property and how do we get that, reductionist biology will not give the answer. It is at this time that biology entered the third phase. The first phase was organismic biology, where taxonomy, systematics, ecology, evolution, all these are investigated. We can call it in vivo biology. Then we got reductionist biology from 17th century onwards. Last 300 years, we are following this. This is, you can call it in vitro, ex vivo biology, but it has a deficiency. It does not explain emergent properties or phenomena like mind, evolution, development, etc. And then we are now in systems biology, which is the theme of this refresher course, which is so-called web of life, and that is a truth. Gaia hypothesis essentially says the entire earth is a living organism, single living organism. And so if we understand the properties of living organisms and the definition of living state, then we understand that the only way to understand biology is through systems biology. And I will explain this with one example from our own area, and that is endocrinology. Now let us look at this. Living organisms exhibit a high degree of order in structure and function. We already said thermodynamics predicts that there cannot be order in this universe, but living organisms have an extraordinary order. The very existence of life has a low probability given the physical principles of thermodynamics, which reveals the fundamental behavior of universe as moving towards disorder, entropic doom. And therefore, Cyril Ponnamperuma, an exobiologist from Sri Lanka, but he worked in USA in California, the chemistry of the universe seems to be leading to life. The universe seems to be in the business of creating life. Therefore, producing life and evolution of life is something built in the structure of this universe or nature. Darwin's idea of evolution by natural selection is the greatest scientific concept in human history. It pervades the whole of biology as a means of interpretation of data and as an undercurrent storyline. Biology is essentially a historical science. Biology is the story of life on earth. Biology is a historical in nature, and when we teach biology also, most people teach it as a series of events giving calendar dates as who did what and what year. And this reveals the story of life on earth, and that's how biology should be taught. However, if you look at it from physics perspective, energy input and communication among components as well as between organisms and environment keep the living organism in a steady state where all physiological and behavioral processes occur like a well orchestrated symphony and not as an individual music. And this is the crux of the web of life or systems biology that we will talk a little bit. Therefore, this organized orchestra, either within a given complex organism like a higher vertebrate or a plant, or environment and population interaction, essentially this orchestrated living activity is possible only because all living organisms interact with each other, and for a given community, different populations interact with them, with each other, and within a population, all the members of that species interact with each other. And for an individual, the individual interacts with the environment, and within the complex individual, all the tissues communicate, cells communicate with each other. And therefore, basically, there is a web of life, whether you take one individual and functioning in physiology as an example, or you take ecology as an example, or you take whole biosphere as an example, what you see is a web of life where constant interactions between the members of that leads to an emergent phenomena which we call as living or evolution at the higher level. And that is the essence of biology. And I'm sure no biology teacher teaches these things because they teach it as information package. And that's how taxonomist will not talk about biochemistry. Biochemistry fellow will never talk about taxonomy or other things. This is unfortunate, and this happens only in this country because people start separate departments only to teach fragmentary information and not with the purpose of understanding biology, and therefore make students understand biology. That was never the aim of biology teachers. Very unfortunate. According to Maturana Varela, these are Latin American Argentine biochemists and philosophers. Fritz of Capra is a physics fellow. Living organisms, watch carefully this definition. This is self-replicating, self-referring, self-regulating, autopoietic energy systems. It takes a lot of time and study to understand this definition, and, and this is essentially the biology that we have to understand. How do they self-replicate themselves in reproduction? How do they self-referee? We are aware of ourselves. We constantly look at the environment and correct ourselves. This is called self-referring. Self-regulating, because our internal system, in the very first day, first lecture, Professor Manohar Achari talked about homeostasis along with evolution genetics as something characteristic of biological systems. And that is correct, because there is a tremendous amount of regulation that goes on within organisms, between organisms and environment, and so-called physiological ecology at the higher level. This regulation is also something that we have to understand, and for that we have to understand only through web of life or systems biology approach. Now, taking endocrinology, which is what I do research for a long time, we will take this as an example and tell you that endocrinology can be taught as a part of a descriptive biology, the so-called phase 1 biology, where you describe endocrine glands, their histology, their structure, and forget about their function. And it can also be taught as an example for reductionist biology, where we talk of a list of hormones, how much is there in the blood samples, what do they do, how do they act on target tissues, what is called as mechanism of hormone action, and leave it at that. But that also does not allow you to understand complete endocrinology. We have to deal with endocrinology as a systems biology approach. I will show you some examples for that. Then we will try to understand emergent phenomena in the field of endocrinology as an example to biology. All right. And this interaction among the tissues and cells of our body, I am talking a higher mammal as an example. This is true for any other hormone, for any other organism or any other living organism. This interaction is through communications, and that is the crux of web of life. Without communication, there is no interaction. And communication is characteristic feature of internal physiology as well as external behavior or even development. This communication is simply a codified message. You see this in the operations of the army while fighting an enemy. They communicate with each other. And this is always in a code, and all communication is in a codified message. Hormones are also codified messages. It takes place, say, for example, among organisms. The most evolved of these is human language, which is a communication mechanism. But we will not discuss this today because, for many reasons, not the least is that man has culturally evolved now, and so he is clever enough to use language to hide his thoughts and not to convey any message. This is a very philosophical interpretation of how culture and civilization has corrupted human mind, and therefore communication is used only to hide, not to express. We will leave at this. Evolution of languages over time from a comparative linguistics perspective reveals the story of human migration. I told you that biology is the story of life on earth, but nobody seems to teach you the story of human migrations and the human evolution. For a simple reason, you have to study comparative linguistics to understand these things. But this evolution is not due to selection pressure. and hence we will not discuss this here. Now, the communication that I am referring to in a vertebrate higher mammal inside our body, there are three types of communication. The first type of communication is between or among organisms of a group. This is called a population, as most of you are aware. Or between species, there are in behavioral ecology, you will be told about communication between species also. It could be either intentional or unintentional and results in influence or effect. We will not discuss this much because this is the type 1 communication. The type 2 communication is between the inanimate and animate world. Please remember, ecology tells us that we have to look at the interaction of organisms with the habitat, the physical habitat, and therefore that only gives us an understanding of behavior. So this is the communication between inanimate and animate world. I do not like to call it even communication, but for want of any word, I am using that. It is influential because we know organisms respond to environments like photoperiod, temperature, rainfall, monsoon, all sorts of things. It is not intentional. So when it rains and somebody responds with the reproduction activation, it is not intentional. It is automatic. It is a what we call instinctive action. Photoperiod effect on living organism is a classic example. And the third type of communication is between the cells within the organism or tissue. It is not intentional, but results in the regulation of a set point. This is the homeostatic set point, which Professor Manohar Acharya was referring to in the first lecture. Let us understand this a little better. We refer to a system of signals. Communication is through signal. I have already mentioned. We refer to in endocrinology as a system of signals operating within a higher animal or plant, and which is essentially intercellular communication. Signals are mostly chemical, but occasionally physical. I will give you a list and we'll see this in more detail. We will discuss the nature of these signals, their origin and transmission, their reception and decoding, and how they bring about control, regulation, and order within the living organism, essence of biology of individual biology or organisms. These signals are of basically two types. Some are physical type, light, pressure, and voltage. You are all aware that photoperiod affects all living organisms. Entire photosynthesis in higher green plants essentially is an influence of light on the biochemical processes within the leaf or green leaf. Therefore, animals also respond through photoreceptors which are present in pineal gland and at other places. And therefore, light is a signal, a communication between the environment and living biosphere, and we respond to this, and therefore, this is a regulatory signal. Pressure is a mechanical type. Our hearing, when we hear music or anything, these are essentially signals in the form of pressure changes in the air, and we respond to this. Therefore, pressure also is a signal, change in pressure. Voltage is a signal. Our entire brain neural activity is electrical activity, electrical in nature, where voltage differences across the membrane, and therefore they are called excitable membrane. These are serving as communications through neurotransmitters, but the initial event is essentially electrical in nature. Voltage difference across the plasma membrane. Many signals are chemical type. I refer to in endocrinology hormones, which most of you are aware of. And these hormones are chemicals in nature, in chemical nature. There are neurotransmitters in animal systems where a neural system is also endowed with, and there these neurotransmitters are also chemical in nature. In addition, we respond to many chemicals which come from outside in the form of drugs or plant extracts or any other thing, and those are also, they are called as xenobiotics, and we respond to them. They also serve as signals. That's why you see these hallucinogenic materials from many hallucinogenic plants having effects on behavior, human behavior, animal behavior, and all. Therefore, we include them as signals and the ability of human beings and all living organisms to respond to this. This is called sensory perception, and most of the textbooks will tell you that we have five senses. A sense of sight, where light mediates this, sense of sound or hearing due to mechanical changes in the air pressure, touch, which is also a mechanoreceptor mediated, taste, which is a chemoreceptor, therefore chemicals serve as signals, smell, which is olfactory signals, these are also due to chemicals. But what your textbooks don't tell is that we have seven more additional senses. We have a sense of direction, which is called magnetoception. If you see animal migration in Kenya or Tanzania or any other place, or even bird migration and all, you will notice this is an extraordinary sense of direction. When Siberian birds come to Bharatpur near Delhi to the same tree coming across 10,000 kilometers, this is an extraordinary sense of direction. We have a sense of pain. This is called nociception. There are lower forms of organisms which don't feel pain because pain is a property of neural system. The pain is felt in the brain, and therefore this is only found in some set of organisms, not all. We have a sense of balance. Many people can walk on a big rope without falling, while most of us cannot do. This is a sense of equilibrium, and this is called as equilibrioception. We sense acceleration. If you are going in a train or a motor car, whenever there is an acceleration, you feel it. This is called kinesthesioception. And the most important and the most puzzling is that we have a sense of time. This is called Zeitception. Zeit is a German word. Now, we also have a sense of space. When we enter a room or a building or a cinema theater, we understand immediately how big or small it is. Now, that means we can sense the volume or size. We can also sense the lapse of time. As most of you know, we have memories, and that will tell us what is past, what is future, and what is present. We also can have a sense of number. Microbiologists talk about quorum sensing, and this is also a part of the additional senses that we have. Now here, the most important thing is that means living organisms have a sense of time, sense of space, sense of number. Mathematics developed only because of this sense of number. But it is still a big puzzle. How do we measure time? How do living organisms measure time and space? It is still a mystery in biology. But no biology teacher will tell you what are the mysteries of biology, what are the questions of biology, and what are the questions which are answered, and what are the unanswered questions in biology. Because biology is taught in fragments as information transfer, as botany, zoology, microbiology, or as biochemistry, genetics, physiology, or molecular biology. And therefore, you will never hear the full excitement of biology unless you are taught biology as a single conceptual discipline. All right. Some chemical signals that I refer to in endocrinology, we should know the chemical nature of these. Some of them are amino acid derivatives. Thyroid hormones are amino acid derivatives. Steroids, sex hormones are steroidal in nature. Lipids, prostaglandins, and all pain mediators, they're all lipids in nature or fatty, long chain fatty acids, isoprenoids. There are many signals which are sugars. For example, you have read in molecular biology that there is something called lac operon which operates in E. coli. Whenever E. coli sees external lactose or glucose, it will sense it and mount a response. And that therefore sugars are signals. There are gases like nitric oxide, for which a Nobel Prize was given, hydrogen sulfide, which also act as signals. Most of our cells are regulated through metabolic changes in nitric oxide. Your endocrinologists and others must have told you about all this. Plant systems, till today, they have not discovered a typical nitric oxide synthase enzyme, but they're searching for it. These are the aspects which biology teachers should teach as unifying concepts in all areas of biology, but should not teach only as botany or zoology or something else. Nucleosides are also like adenosine, serve as some of the plant hormones. For example, isopentenyl adenosine is a hormone, and that is essentially a nucleoside or a nucleotide derivative. Therefore, we summarize, when you look at all living organisms from this perspective, signals regulate everything in physiology, development, and behavior. Physiology, development, behavior are the living processes, whereas structure description, morphology or anatomy or internal anatomy, histology, they are only descriptions of structures. When we relate structure to function and raise an important question, the most important question in biology, which no biology teacher ever explains, does structure subserve function? This is the theme of biology which we should teach, and this comes under systems biology. And therefore, it is the signals that mediate this regulation, and that endocrinology is the grandest example for this, and developmental biology. And I mentioned that in higher animals, this is through neurotransmitters, through a system called nervous system. It is preferred to call it neural system. And all of you are aware of all these things. I don't have to go into detail. The endocrine system, which was officially discovered in 1839, but the historians in your textbooks will only talk about 1905, when Starling discovered duodenal hormone in a rat. And from that day, endocrinology was born. However, this endocrinology, if you look at it, essentially as a part of biology, asks and answers three questions. Does structure subserve function? I have already mentioned. What are the mechanisms of hormonal effects? Because I said all these links or signals are called hormones, and therefore biologists, endocrinologists asked a valid question: how do hormones bring about this? I'll give you some examples. Then you see the mystery behind hormone action. Whether it is a plant hormone or an animal hormone, it does not make any difference. What is the signal and how is it regulated? In spite of 120 years of endocrinology, we still don't have answer for the third question, and that is the unanswered problem in endocrinology and in biology, regulatory biology. Now I'll give you some examples of these endocrine glands and the hormones they bring. Pituitary gland is something that is found below the hypothalamus, below the brain. In the brain, if you break the skull and scoop the brain out, you will see the floor, and there is a thin membrane there, sella turcica. With a scalpel, if you remove that, you will see pituitary gland as a small 1 or 2 milligrams in a mouse, but about 1.2 grams in a buffalo. And this pituitary secretes a number of hormones, and all of these hormones regulate the so-called peripheral tissues. Here is an example. A hormone called prolactin regulates mammary gland or breast, both structural development, differentiation, and secretory function, milk production in mammals. ACTH, adrenocorticotropic hormone, regulates adrenal gland. It is also called as suprarenal gland. Thyroid, thyroid stimulating hormone from pituitary regulates thyroid gland. Growth hormone regulates bone and cartilage. Then there are hormones called gonadotropins, which regulate gonadal development, differentiation, and maintenance of function, like gametogenesis, spermatogenesis in the male, oogenesis, and ovulation in the female. And these are done respectively in the female gonad called ovary and the male gonad called testis. We did not go into detail all this. This comes under reproductive biology. So when the reproductive biology teaches reproductive biology, he will not talk regulation because he doesn't know other areas of biology. That is the tragedy of biology. And then there are other hormones which regulate kidney, antidiuretic hormone, uterus through a hormone, peptide hormone called oxytocin. These are only examples. Signals originating from hypothalamus and pituitary are chemical in nature. They belong to a class of chemicals called peptides and protein. So we are now talking about the reductionist biology, a little bit more on chemistry side. Here is another example where a pituitary or a hypothalamus produces a hormone called GnRH, gonadotropin releasing hormone, a decapeptide, and this acts on pituitary gland. It is delivered through nerves, and this pituitary receives them from the extracellular compartment, and in response to this specific hormonal signal, gonadotropins, LH, FSH, are released from the pituitary, anterior pituitary, called adenohypophysis. Similarly, posterior pituitary secretes other hormones like oxytocin, vasopressin. Now the point is, in response to this, when pituitary secretes its own hormones, growth hormone, prolactin, ACTH, I have already shown you the list, they in turn go through the blood and act on peripheral tissues. Here is an example of a growth hormone acting on liver and then mediating secretion of insulin-like growth factor, which act on other tissues, and therefore the body grows. And therefore, this is an example of what we call the spectrum of actions of hormones from different tissues, but acting on each other as signal, and the responding tissue again responding to that through metabolic alteration. But what you see is an emergent phenomena, emergent property. In this case, it is growth. Okay. Same thing for gonadal activity like gametogenesis, reproduction. It is a emergent property mediated by signals of respective hormones. This is another example. Thyroid stimulating hormone, releasing hormone from hypothalamus mediates, acts on pituitary and releases TSH from pituitary. And the TSH acts on thyroid gland. In turn, the thyroid gland releases T3 and T4 or thyroxine and triiodothyronine. But what you see in this diagram, that the products of the peripheral tissue act on the source of the inducing hormone. In this case, I have shown T3 and T4 acting on pituitary and inhibiting the release of TSH. And this is a feedback inhibition. Now you see that the way signals and act in our body, it resembles an electronic circuit. These are called feedback loops, feedback inhibition, and so-called feed-forward activation. Most of you have houses and you have circuit breakers in your electric supply scheme. That means whenever the voltage is high, you see the circuit breaker operates and cuts off the current or the electric supply. Same principle is operating in our endocrine system, and therefore we say that the endocrine signal system works like the principles of signal engineering, and that is where a systems approach will make us understand. It is same thing here shown in another way. Feedback inhibition and feed forward activation therefore are characteristic of signaling mechanisms in our body. There is a network of signaling. This is the language of systems biology, or so-called um, third phase of biology, and metabolic pathways with built-in feedback loops which regulates living processes. This is the summary of endocrinology from the so-called in silico biology approach, where modeling is done and where we have understood that the system of signals or communication between cells, among cells, results in some regulation of metabolism, and therefore an emergent property or a phenomena called secretion happens. And therefore endocrinology is a grandest and the best example to study this web of life within a given organism. At ecological level also, this is operative. Now what we have missed in this narrative is that a signal is as good as only if you can decode the signal. As I said, supposing I am a deaf person, any amount of music by Lata Mangeshkar or Mohammad Rafi has no meaning for me because I cannot hear. And therefore, music makes sense only if I have a reception system. And people have studied through reductionist biology, biochemistry, and molecular biology that this reception is mediated by biomacromolecules called receptor. Therefore, receptor is a pharmacological term. It refers to the entity in the target tissue or an animal or an organism which responds to a signal. And this has three functions: signal discrimination, signal transduction, signal amplification. That means if I am listening to Lata Mangeshkar very closely, any amount of noise that you make, a dog's bite or a car horn, I won't listen because I am carefully listening to Lata Mangeshkar, which means this is called signal discrimination. We exploit the signal to noise ratio and pick up only the signal and not the noise. The second property is signal transduction. I will give an example and then you will understand. And the third property is that signal amplification, which happens because by definition, signal is a coded message and hormones concentrations are very low in nanomolar and femtomolar range. Therefore, the signals are hardly whispering, but the tissues will listen to them loudly like a loudspeaker, and that is done by the tissue in the form of signal amplification. When we take up an example, we will see. Now, these signals therefore act on target tissues, and there are two types of responses or activities, actions. One is called acute action. If you take an injection of insulin, you will see your blood glucose level comes down within 1 hour. And this is therefore acute activity or an example for acute action. On the other hand, there are actions which are called chronic actions, where it will take not just 1 hour, it will take some days and weeks, many times years, and then you see an emergent phenomena coming out. Growth is an example of this. Development is an example, or embryology or any tissue differentiation is an example of this emergent phenomena, but it is a chronic effect, long-term effect, done, initiated by the same hormone or the signal. And therefore, there are two types of signal responses: acute response and long-term chronic response. And chronic responses lead to emergent properties. Acute response leads to metabolic changes. When you measure blood glucose, it is a biochemical experiment. So it is very simple and easy and clear. But when you measure growth or development, we are into trouble because you don't see what is happening behind this and which results in this growth. This is an example where I have shown you in the slide. If you inject less than a microgram of estradiol 17 beta, a female sex hormone, into a immature or a castrated female rat, you will see that the size of the reproductive tract, uterus, increases enormously. In the middle, you see the animal which has been castrated, and in the left side and right side, you see the increased uterine size because of the hormonal administration. And this is a dramatic effect. After all, when all of us watch a young child grow up into a handsome man or a beautiful girl, woman, it's a marvelous phenomena, but nobody believes that this is an emergent phenomena initiated and maintained by nanograms of hormones as signals. And that is the beauty of systems biology. Similarly, in the male, another example I have given in the influence of androgen, accessory sex glands, one example is given here, seminal vesicle. In a rat or a mouse or an elephant or a man or anywhere, it enlarges in size, and that is shown here. Essentially, therefore, a system of signals starting with the environment, going through reception systems at multitude of tissues, results in acute actions and thereby regulation of the metabolism in the target tissue, as well as chronic action or leading to emergent phenomena where homeostasis is maintained as well as emergent properties or phenomena occur. Entire evolution is an emergent property or a phenomena occurring due to underlying living processes in individual populations. Similarly, behavior is an emergent phenomenon. Now, there we will see the distinction between mind and brain. We will not deal with that now. Let us take another example of an emergent phenomena. A newborn baby suckles her mother's breast, and you will immediately see milk let down from the mother's breast. What actually is happening is it's a neuroendocrine arc where the suckling stimulus stimulates secretion from hypothalamus as well as from neurohypophysis, and that comes in the form of a hormone called oxytocin, which and prolactin, of course, and oxytocin results in contraction of the breast muscle, and that results in the secretion. And this whole thing is orchestrated by a single stimulus of suckling, and the entire underlying living processes, biochemical events, are orchestrated exactly in an ordered fashion. This is why we say, how do you get milk out from a biochemical event? This is the emergent phenomena coming out of living processes or metabolic changes. All right. This is another example, ovulation. All mammals or any sexually reproducing organism will lay eggs, and this egg release is called as ovulation. And there is an underlying series of activities, biochemical in nature, initiated by a single hormone, luteinizing hormone, and that results in this, which is a complex phenomena. People have taken 100 years to understand the details about this process, and this is an emergent phenomena example in reproductive biology. Same thing for other thing. Some of these hormones are not always peptide or protein. They are also steroid in nature. Now we see a marvelous emergent phenomena many places, but here is one example. In the case of amphibians, if you study the life cycle, you notice that there is an intermediate form which they call as tadpole, larva as it is called. And this insects also, you will see this kind of thing. Now, if you see this, the fertilized egg, fertilization incidentally is external. Initiated ovulation release is spawning, it is called. It is released by hormones. And this fertilized egg develops, differentiates into tadpole. It lives for some time and then undergoes a remarkable differentiation, multiple activities synchronously happening, where it transforms itself into a frog. Now, this event, which you can write a whole book on this transformation and the underlying biochemical changes, is initiated only by a simple few nanograms of thyroxine as a hormone. And that is the miracle that you see here, a mystery. How does a frog come out of this tadpole by simply nanograms of thyroxine? We will take another example of emergent phenomena, and this is health. Most of you must be wondering, what is health? You search, read all books, there is no definition. Nobody understands. Worst of all, World Health Organization, the leader for maintaining world health, it also has no understanding of what is health. It gives definition of health as physical, mental, and spiritual well-being. Now, in science, we have not defined what is mind and what is spiritual. These are not scientific words, but WHO uses this. Anyway, but we all understand what is unhealth. Any discomfort is called unhealth. In human history, different societies have interpreted health and unhealth differently. The four humor idea of Hippocrates, the Panchabhuta and Tridosha ideas in Ayurveda, the ideas of chi, prana, and pneuma in Chinese, Hindu, and Greek thoughts are some of the examples of what constitutes our health. Indians have talked about health in a different way. You must have heard of placebo effect, which is a biological effect that you see by essentially mind acting on the body. So this placebo effect, the mediation and the details about it tells you that mind has an influence on brain and other body activities, and therefore mind is external to brain. And that gives rise to a philosophical problem which physics does not answer, and that is that biological processes, emergent phenomena are directionless. Your evolutionary biology teacher would have told you that there is no direction in evolution. Similarly, here you see a directionless event occurring, and therefore we believe biology in philosophy terms says that we have free will. It is not talking in religious terms where they said everything is due to fate. What is written on your forehead? This is all bullshit. Absolutely, we have free will. That is what biology teaches us. Physics gives you an idea of universe which is deterministic in model, cause and effect. Biology does not talk about it. There are only correlations here. We don't know what are the ultimate causation. And therefore, this placebo effect is an example of where everything is a mystery and biology does not answer all this. Same thing, in biology, we say health is a balance between mind, that is brain, endocrine system and immune system. Without going into detail, it shows that this is a homeostatic condition, health, and we have to understand it. Behavior is also an emergent phenomena, and this is also induced by hormonal signals. Quickly, I will go through this example. Serotonin gives you a sense of unfairness. This is a real-time experiment, and it is published in Science. A person went to rob a bank, and while coming out, another person watched him and said that I will give a complaint to police that you are robbing. Immediately that robber tells, I will give you 10% of my robbed money. Don't complain and be set, keep quiet. That fellow says yes and is happy and does not make a complaint. The same experiment was repeated when this fellow's observers basically were given a 1 week diet without tryptophan, an amino acid, and tryptophan gives rise to or biosynthesized to serotonin, which is a neurotransmitter which affects behavior. And when such a man is now repeated in the experiment, where he observes this robber and the robber says, I will give you 10%, he is getting angry. He is not happy. Then he says 90%, 80%, then he agrees. And therefore, sense of unfairness is because of serotonin. This is a marvelous example of so-called reductionist biology. But you see, it is explaining behavior through molecular changes. Like that, we cannot, we can go on, but I will end up here by saying we do not understand how anticipatory changes, signals and signal actions occur during development. There are many examples, and I have already given you amphibian metamorphosis as a greatest mystery. How do living organisms measure time and space? Because all these events are orchestrated. Here is another example. This is a penguin bird. The newly hatched penguin bird, in about 3 months, it has to jump into lake or Antarctic water, which is cold, extremely cold, freezing. And as though it is going to jump 2 months later, it develops a thick lipid layer on its ventral side. And this is anticipatory changes in gene expression and biochemical changes. And we don't understand how does the chick know that it is going to jump into cold water. These are called anticipatory changes. These are also told in genetics as programmed changes, but we don't talk program. It's only runs a program. It is a potential. It can be bowled out. Accidents can appear, and therefore we don't understand how this ordered action, and which is also sensitive to intervention, happens in animals and plants. And that is why the mystery of the so-called anticipatory actions. We can go on like that, many examples. I will stop at this. Today we can reprogram cells also. All developmental patterning is basically due to signals, and these signals give rise to patterns. Information transfer through signals, receptors, and feedback loops, characteristic of systems biology, so-called web of life within an organism, mediate all physiological and behavioral processes, achieving coordination and health. Defect at any step results in unhealth and also suggests therapeutic approaches. I will end here by saying that I have taken endocrinology as an example to teach systems biology or the web of life and its implications. But luckily, endocrinology is also an example for teaching reductionist biology, which means biochemistry, biophysics. It is also an example to teach class 1 or classical biology, where we can talk about morphology and histology. Therefore, biology should be taught as a single conceptual discipline, not as fragments, not as information package, as it is presently being done in majority of university departments and colleges. We are committing crime in teaching fragmentary biology and not total biology, because we'll never understand except if you teach biology as a conceptual discipline. Now, I also feel uncomfortable in addressing computer. Therefore, I am showing you this slide where it also says, let us meet on the old watering hole. I would have been much more happy using a blackboard and not this computer online education. Thank you very much for your patience. I can answer any questions that you have.
Sir, there are two questions. Really, sir, it is wonderful. Please, sir, tomorrow's lecture you start with this illustrations, what you skipped today. Really, it was, we say that serotonin story, it was wonderful, really. And tomorrow, please, you continue from the one which you have missed today. I hope you will do. There are two questions, quick questions I'll ask by the participants. What does in silico mean?
Okay. In silico means, as I said earlier, biology developed or grew in three phases. In the first phase, which lasted about 1700 years, starting from Aristotle, it was only description using naked eyes, pair of eyes, later magnifying lens, later microscope. You went on describing what you see in nature, and that is called descriptive biology. It is also called phase 1 biology or classical biology. We got great disciplines like taxonomy, systematics, and it was revolutionized by the concept of evolution, organic evolution by natural selection, Darwinian idea. Later on, we practiced what is called reductionist biology, biochemistry and biophysics. I have already described all this. For 350 years, we are doing this. But both the classical biology and the reductionist biology did not lead us to understand. And therefore, people said, stop doing all work, be intelligent, buy a computer, do modeling by all the data that others have taken, you take it, put variables, study the interactions of these variables among themselves by differential equation, and therefore use mathematics to construct modeling of a given phenomena like menstrual cycle or behavior or anything, climate effect on agriculture, anything you can do. This is modeling, and this is so-called details of web of life understanding through computer. And because computer is called in silico, it is called in silico biology. So we have in vivo biology, which the taxonomists and evolutionary biologists teach, morphologists teach. We have in vitro biology, which the biochemistry fellows, biophysics fellows, physiology people teach. And we have in silico biology, which the computational biologists teach. We should teach all the three. That is the danger. None, no department, no university, no college teaches whole biology, and that's why we are in a sorry state of our affairs. Biology, for me, is the most exciting subject, but teachers have made it the most boring subject. Did I answer your question?
Yes, sir, it was a very good description actually, your expertise. One more question is, what is philosophy of reductionism?
Yeah. See, philosophy in the old name for any knowledge. Okay. Now, slowly, this knowledge became developed into different branches like physics, chemistry, biology, history, geography, and all that. And therefore, slowly, philosophy could not manage analyzing all the areas of human knowledge and restricted itself to certain specific set of questions. And therefore, we have a philosophy, one which asks only what is beauty. If you see Greek philosophy, it asks only two questions. What is ethics? And therefore they developed an area called aesthetics. What is beauty? Ethology and aesthetics are the two areas of knowledge which developed by philosophy of Greek philosophers. But in the so-called Middle East, as well as in East China and India, the philosophers asked not about nature or beauty or anything. They asked about what happens if I die. So therefore, they brought the hypothesis of God. Great religions were established. They manufactured God and said that established religions. So if you see the three Abrahamic religions, Judaism, Islam, and Christianity, they are basically essentially a philosophy which looks at the relation between dead people, wherever they are, and living people. And so great religions came, including Hinduism and many other things. However, science, as I mentioned, deals with only the nature. It does not act or deal with supernatural things like God or anything. And therefore, this science had its own philosophy. And this philosophy of science till today is written mostly by physics professors. And therefore, you have certain belief in certain philosophy of science. However, biology, through some new questions, where we had to write a new philosophy of science through biologist's angle. One of them is reductionism, where we say a whole is equal to sum of the parts. I will put it in arithmetic. 2 plus 2 is 4. I don't think anybody will fight with me or throw stones on me if I say this. Essentially, reductionism says we can understand biology, biological processes, structure and function if you simply reduce this biology in the language, in the techniques and tools of physics and chemistry. Therefore, if you reduce biology to the biochemical and biophysical language, that subject is called reductionist biology. However, there is a problem with this biology. Although biochemistry people are very proud of their subject, I am also a biochemist. Biochemistry will not lead to the understanding of biology because it does not explain emergent phenomena. I have already talked about it. But this philosophy that reductionism reduce it to chemistry and physics will explain biology is a philosophy of biology, reductionism. However, we know biology has emergent phenomena. Now I will put it again in simple arithmetic. In biology, 2 plus 2 is 5, not 4. That extra 1 is the emergent property. That is the explanation of reductionism and the trouble with reductionism as a philosophy. Did I answer your question?
Participant, yes, sir. Participant is very happy. He said very nicely explained. Thank you, sir. He said, thank you, sir. Sir, can you stop sharing your screen because next speaker is already ready?
Yeah, sorry. I'm sorry. I can also go up. Yeah.
Okay. Thank you very much, sir. Again, once again, you will be meeting. We will meeting you tomorrow again in the morning.
Yeah.
So, sir, please you start with the illustrations. What we have skipped today. Such a wonderful story of the thief and the experimenter. Good morning, sir. Good morning, Insar, sir. So it is a proud moment for us. We are delayed by 10 minutes. It's hope. Kindly regret for that. We are today with us Dr. Insaf Ahmed Qureshi. He is a senior grade assistant professor from Department of Biotechnology and Bioinformatics, School of Life Sciences, University of Hyderabad. So he is masters from Jivaji University, Gwalior, in applied microbiology, and he has his PhD from Hamdard University, New Delhi. His PhD thesis was isolation and characterization of lectins and their genes from chickpea. And his research experience is presently is working as assistant professor in University of Hyderabad. He worked as visiting scholar in Department of Biochemistry, University of Missouri, Columbia, USA, from December 2013 to 14 and December 2018 to 19. He worked as postdoctoral research fellow in Division of Structural Computational Biology, School of Biological Sciences, Nanyang Technological University, Singapore, during 2007 to 2010. He worked as project associate in Structural Biology Unit, National Institute of Immunology, New Delhi, during 2005 to 2007. Honor and awards to his credit are: he was selected as esteemed ICMR International Fellowship for Young Biomedical Scientists during 2018 and 19 by ICMR New Delhi. He was awarded Indo-US Raman Fellowship by University Grant Commission New Delhi for year 2013 and 14. He was selected for esteemed international exchange program by INSA New Delhi during 2013 and 14 and 18 and 19. Awarded for ICMR Prize 2010 by ICMR Delhi for year 2010. Awarded with Fast Track Project for Young Scientists by CERD New Delhi 2012. Associate Editor in Gut Pathogen Journal during 2013 and 17 with impact of about 3. Editorial Board Member of Current Protein and Peptide Science Journal from 2021 onwards with impact above 2.5. He has number of publications to his credit. Near about he has 60 publications and number of structural coordinates in protein data bank, and he delivered several lectures in symposia like in 2020 in our Telangana University international conference. He has delivered a wonderful talk on development of novel multi-epitope vaccine against SARS COVID to using immunoinformatic approaches. He delivered a talk on structural study of Leishmania proteins in refresher course in life sciences at UGC Academic Staff College, University of Hyderabad during November 2020. He delivered a talk in 89th annual session of National Academy of Sciences India at ICRNA then AARM Hyderabad on biochemical and structural attributes of pyridoxal kinase from Leishmanial parasite. He delivered a talk on protein structure visualization in workshop on molecular technology tools and techniques in NIT Warangal on 23rd of October 2018. He delivered a talk on determination of protein structure in workshop on basics of bioinformatics for PG students at Mahatma Gandhi University, Nalgonda in 2018. He has delivered a talk on laboratory practicals in bioinformatics and refresher course in life science at UGC Academic Staff College, University of Hyderabad. He delivered a talk on visualization and validation and analysis of structure in workshop on protein structure and drug discovery at University of Hyderabad. He delivered a talk on model building and refinement in workshop on protein structure and drug discovery. And likewise, he has delivered a talk on protein crystallization. He delivered a talk on computational biology applications, recent advances in biological sciences. So number of refresher courses, he has delivered wonderful talks and motivated several people, teachers and other participants. To his credit, there are a number of projects, like ongoing projects. He is having a very big project from ICMR in vivo validation of membrane acid phosphatase as a vaccine candidate against leishmanial parasites. Also, he is having an ongoing project from CERB, structural and functional studies of glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase to understand their role in leishmaniasis. He is also having one more SERP project, identification of novel alleles of wild rice derived bacterial blight resistant genes and their functional analysis. He completed number of projects like from CSIR. He had completed a project in April 2020 understanding the role of aminopeptidases in leishmaniasis through structural and functional studies. Also, he had completed one more project in 2016, elucidate the biochemical role of protein-protein interaction in the post-transcriptional regulations. Also, he has completed a project in 2015 from SERB, expression and purification and crystallographic studies of protein kinases from Plasmodium falciparum. And also, he has completed a project from UGC in 2015, identification, purification, and characterization of protein phosphatases PP1 from Leishmania donovani. Also, he has completed in the same year a project from UGC, identification and characterization of XA21 and XAL3, XA13 alleles conferring disease resistance to Xanthomonas oryzae from wild species of Oryza nivara. Also, he has completed one more project in 2013 from the University of Hyderabad grant, crystallographic study of casein kinase as a structural based target against malaria. He has number of scholars. He has awarded number of PhD scholars as a research supervisor, like Santosh Gatreddy. He worked on understanding the catalytic mechanism of ribokinase from Leishmania donovani using the structural and functional approach. Also, he had got awarded in 2020 Sayana substrate specificity and structural analysis of paradoxal kinase from Leishmania dorovani. Salim Yusuf Bhatt has submitted his thesis on characterization of peptidase T and methionine aminopeptidase 1 from Leishmania donovani. And ongoing research scholars with him are working on various concepts of Leishmania donovani, and he is a member of various scientific societies like National Academy of Sciences India, Indian Crystallographic Association, Indian Biophysical Society, Society for Plant Biochemistry and Biotechnology, Telangana Academy of Sciences. So, with this introduction, I would like to request Sir to start his presentation. Good afternoon, Sir, and welcome to our refresher course of 14 days supported by Science Academies. Over to Insaf Ahmed, Sir.
Thanks, thanks to Dr. Halim Khan. I'm highly thankful for your detailed introduction, actually, and I'm grateful to you that you have invited me again. Actually, that yes, I have given a talk actually in the month of the, I think, November actually, and you have conducted another program. Okay, so just before starting, I just want to tell to students, like to my colleagues, to be very friendly in this and to be interactive, because this is not a just now recently you have come out from a great talk actually from Professor Murlidhar actually, and he is because he's also part of our university, so we have listened him several times, and you can understand like the way he present and the way he's having things very clear. So I'm pretty sure like you are still in that mood actually, like you are expecting same thing. I will try my best actually, and this one will be more because his talk was basically more theoretical, and here I will try to be more give you hands-on actually. So my request to be you try to be more interactive in this class actually, instead of listening and I'm just speaking and you are listening. That will not work for both of us actually. And here I think maybe in the schedule even Dr. Khan has mentioned clearly like this is kind of hands-on. So in this I will firstly tell you some basics, and then I will try to show you some softwares which you can use and apply in your research. And like you can see in like I have published something actually, some small work actually using all these tools, whatever I am going to share with you. So because and I understand like all of you are from different universities and colleges, so when you will go back, because refresher course is always like I am also assistant I was also assistant professor and still I am assistant professor, and I can understand the pain which we face during the refresher course. So so I don't want to make it a, like a just I will pour my information. No, I just want to discuss with you, and I want to my feeling is like because I am having 6 classes, so I will try to give you something like you can when you will go back to your um, lab or your classroom or your even you sit on the computer, you will feel why I cannot do all these things. And yes, definitely, you cannot publish science or nature paper, but this type work. But yes, you can generate a data and you can publish minimum one or two impact factor paper easily. Whatever I am going to tell you in these 6 classes. So first today I am going to tell you about in silico sequence analysis, because just now somebody asked the question, because firstly I kept title something else. So then somebody asked the question, what is the in silico? I have changed my title a little bit. So because then it is going to fit more actually. And in silico now I need not to explain you, because Professor Muralidhar has explained you in a nice way. I can say in the best way, which anybody can explain actually what is the in silico. So today we are going to discuss much about the in silico sequence analysis. So before in silico sequence analysis, just I want to show you my one slide and I want to ask you a question. Like all of you heard about the GMO. Is it whether I am audible properly?
सर थोड़ा सा आवाज आपका ब्रेक हो रहा है, पार्टिसिपेंट्स ने बताया है चैट में।
Oh, okay. I think that is
और आपने स्लाइड शो नहीं बनाया है,
pardon?
आपने स्लाइड शो नहीं किया है प्रेजेंटेशन को।
Slides को नहीं है? Okay, okay, okay. Is it visible, sir, now?
नहीं, अभी तक तो नहीं हुआ। नीचे वाला आपका वो प्रेस करिए ना इधर कॉर्नर में।
Yes, sir. Is it okay now?
नहीं सर,
Here it is showing your screen sharing.
यहाँ नीचे भी है सर एक। हाँ,
Okay. Now I stopped. Now I will share again. Is it visible, sir?
यहाँ नीचे वाला एक है, देखिए परसेंटेज के बाद वाला जो है ना, वो वाला एक बार प्रेस कीजिए नीचे। आप ऊपर भी कर सकते हैं, नीचे भी कर सकते हैं।
Hello, sir. Is it visible now?
सर आप ये जो ऊपर आपका वो जा रहा है, यहाँ पर आप स्लाइड शो वाले में जाके प्रेस करिए ना, ऊपर ऊपर आपका जो अभी कर्सर है,
Yes,
उस
yes.
स्लाइड शो वाले पे जाइए।
New share.
आप जो कर्सर रखे हैं एरो का,
हाँ.
वो स्लाइड शो वाले पे जाइए आप ऊपर। रिव्यू के बाद एनिमेशंस और रिव्यू के बीच में जो स्लाइड शो वाला है उस पे जाइए।
No, this I am pressing new share actually. It
नहीं,
is not
मेरे
coming.
पास कुछ नहीं हो सकता है, आपका पास पावर पॉइंट है ना, तो आप ही कर सकते हैं।
Yeah, no, that I am doing. I am
आप इधर आइए ना स्लाइड शो पे। हाँ यहाँ राइट वो करिए,
Okay, is it
बस
visible
हो
now?
गया, बस हाँ ठीक है।
Okay. So, I'm sorry. I hope all of you are able to hear me.
ठीक है, आवाज आ रहा है।
Yes, yes. Okay, so I'm not going in detail in the about the in silico, but yes, like whatever sir has mentioned, Muridhar sir, actually that in silico means like if you are using computer for analysis, so that is the in silico sequence analysis. So this slide actually just to try to correlate yourself like GMO, genetically modified organisms. So if you see in the market now, a lot of GMOs are available. Okay, like the best example is the golden rice because it contains, it is having the more beta carotene. Okay, which our body converts into the vitamin A. So it is a good source of the vitamin A. Then similar kinds of tomatoes, like you can see tomatoes, like earlier days tomatoes were getting ripened quickly actually, but now they are far better actually. So these are the few examples of the genetically modified organism. So how to get the genetically modified organism? So for that people use a technology that called recombinant DNA technology. In the recombinant DNA technology, what you do actually? You take a gene of interest, like just now we saw in the case of the golden rice. You take the, because you want to express more beta carotene. So you take the beta carotene gene, amplify that beta carotene gene, and take a expression vector. And in this expression vector, after the promoter, you put that gene of interest. Okay, firstly you digest both and uh, then put your gene of interest here, and after putting you ligate it. After ligation, your construct is ready. Okay, now take this construct and put in a expression system. So expression system is, there is different types of expression system. Like some people use the bacterial expression system, some people use the yeast, some people use the insect cells, some people prefer the human expression system. So they are having their own pros and cons. Like in the case of bacteria, your yield will be very high and cost will be very low. But in the case of the yeast, it will be kind of similar. Yield will be moderate, but cost will be less. But if you are using a suppose human cell, your cost will be very high, but yield will be very less. But why people prefer the human cells? Because mammalian cells are good for the post-translation modification. If you are having a prokaryotic protein, then there is not any problem. You can simply technology to get the specific protein, and that protein can be used for the several purposes. Like best common example is the insulin. I hope all of you are aware that insulin is also produced by the recombinant DNA technology. So we are going to discuss how to get a specific protein and how to characterize. So we will start our exercise from the top. Top means gene of interest. So first my question, how you will get the gene of interest? So generally like if I ask what from where you will get gene of interest, you will say you will get from the specific organism. But that specific organism is having that gene of interest or not, how you will get the information. So for that, you should be aware about the biological database. Because if you see, there is several microorganisms and their sequences are already available, and those sequences have been deposited in the database. So those databases are good source to get the sequences of the gene of interest. So let's start firstly. Let's know firstly about the biological database. So biological information of the nucleic acid available as a sequences. But if you talk about the protein, then protein information is available as a sequences and the structure. If you see, sequences are represented in a single dimension because just you will get a like primary like A, G, T, C, G like this. But if you see in the case of the protein, in the biological database. Protein sequences are also available as a single dimension along with the three-dimensional database also. Okay, so that three-dimensional database for the protein is the PDB database. Similarly, a biological database is a collection of data that organize its content and can be easily accessed, managed, and updated. So why we need the biological database? Because just now I mentioned if you are interested to clone a particular gene and that gene is already available, you can extract from there. So first thing, need for the storing and communicating large data set has gone. As you remember now, if you see earlier days, people was using the Sanger sequencing. And if you want to perform a Sanger sequencing, it will take even months, years to get a proper sequence. But now there is a new technology that called next generation sequencing, NGS. In NGS, within 1 week, you will get a data sequence data of the whole organism. So now a lot of data has come, but this data where we have to put it? Because now suppose I have sequenced one new organism and I want to put in a platform from where other people can use and other people can explore and use for their research. So for that we need the biological database. So first thing is the need for storing and communicating large data set which has grown with the time. Make biological data available to the scientists. So first thing, you are storing. Then second thing, others are able to use. That's biological data. And to make biological data available in the computer readable form. So the data should be this way. It is not like only I can explore the data because this is my data. No, if I am submitting in an open platform, then anybody can explore the data. So that should be in a readable form. So there is different types of the biological database. I don't want to go in those details actually. Just I want to divide in this two part actually because whatever the related to our discussion in the these 6 classes what we are going to discuss. First thing is the nucleic acid sequence database and second is the protein sequence database. So nucleotide sequence, if you see, nucleotide database is a collection of sequences from the several sources like GenBank, RefSec, PDB, etc. Experimental results. So how from where you get this nucleic nucleotide database? So nucleotide database comes from the experimental results. Like suppose you have isolated a novel gene, you go for the sequencing and after getting sequence, you submit in the nucleotide sequence database. So that data is basically experimental data. So experimental results are submitted directly into the database by researcher and the data are essentially archival in nature. So if you see the again nucleotide database or any database, they are divided basically in the three forms. One is the primary database, second is secondary database, and third one is the basically is a combined form of the primary and tertiary, primary and secondary database. So we will talk basically a broader way, primary database and secondary database. So if you see in the case of the nucleotide, in the case of the nucleotide, there are three chief databases that store and make available raw nucleic acid sequences to public and researchers. And those are the GenBank, EMBL, and DDBJ. They are referred to as the primary nucleotide sequence database since they are the repository of all nucleic acid sequences. All these three accept nucleotide sequence submission and the exchange new and updated data on a daily basis to achieve optimal synchronization between them. So this is like, it is not like, okay, this will open annually and you can submit that time. If your sequence is ready now, you can submit now itself. So there is not any hurdle. They are very much open. These are primary database as they house original sequence data. So now just now I mentioned basically there is three database. One is GenBank, another is DDBJ, and third one is the EMBL. So we'll go just briefly about the GenBank and other things. Why I'm telling you, because finally I have to take you how to get the data from a primary database or how to get from a secondary database. Just before going there, I want to tell you some brief information about the those database. So you will have the information like how to these database bases are generated, or how that sequences are available on these databases. The gene bank is physically located in the USA and is accessible through the NCBI portal. If you will click on this, you will reach directly to gene bank. It says open access. Open access means free to everybody. Annotate collection of all the available nucleotide sequences and their protein translation. You can not get only the nucleotide sequence, you can get their protein translation also, translated product also. This database is, sorry, sorry. Yeah, so this database is produced and maintained by the National Center for Biotechnology Information, that called also NCBI, as a part of the International Nucleotide Sequence Database Collection, that is the INSDC. It receives sequence produced in the laboratory throughout the world from more than 1 lakh distinct organisms. It has become an important database for research in biological fields and has grown in recent years at an exponential rate by doubling roughly every 18 months. Similar way, there is another database, EMBL. So EMBL is the European Molecular Biology Laboratory. So you can understand this is basically Europe based. It is placed in the USA, this database. Okay, and this is also having a similar way. It also collects the data sequences from the researchers and provide to the public or scientific community to use them. Similar way, there is another database that called DDBJ, DNA data bank of the Japan. So because this is from the Japan, so this is called so this is the this is available only in the this is available in Japan actually, and but yes, this is also available for the public, and it is the only nucleotide sequence database bank which is in the US, in the Asia. Others are in the other countries. Okay, so let's go for the now after the primary database. So these are the three primary database. What is the secondary database? So secondary database basically like primary database because you have performed the experiment and whatever information you have obtained, that information or whatever the sequences you obtained, you have submitted in a database. So that database will call the primary database. Secondary database is the, sorry, this is not moving. So secondary database is basically like whatever experiments you have performed and whatever sequence you have obtained. From there, if you are extracting any information and that you are submitting as a, you are like, suppose you have taken a sequence and you have translated it. And now after translating, this sequence will go, now this sequence, whatever sequence you have obtained, that sequence if you will submit as a translated sequence, then it will be protein sequence. So then it will be basically, it will be secondary. So if you are obtaining any information, many of the secondary database are simply sub-collection of the sequences culled from the one or more, one or the other of the primary database such as GenBank or EMBL. So there are other secondary databases that do not present sequence at all, but only information gathered from the sequence database. So they are the secondary database. Similar way, whatever we are having for the nucleotide, similar way we are having for the protein also. Primary database for protein and secondary database for protein. Now what happens, like just now I mentioned in the in the GenBank. In the GenBank, what they do, they take the sequence from the researcher and they translate it into a protein. So now whatever protein is available there, that has come from the experimental information. So that is called the primary database of the protein. So the primary database holds the experimental determined protein sequence. From where it is coming? Because if you know, generally for protein sequencing, we follow the Edmond degradation method. Edmond degradation method is having a limitation. Like you can, if you want to sequence a protein, you can get 10 amino acids, 20 amino acids, 25 amino acids. But if you are having a 50 kDa protein, you cannot get the whole sequence of the protein, the 50 kDa protein. So what you have to do? In that case, you can take the nucleotide sequence of that, you translate it, and then you use for your purpose. Okay, so the primary database holds the experimental determined protein sequence inferred from the conceptual translation of the nucleotide sequence. Now, this is not basically, this protein sequence has not come from the protein database, or like it is not you have performed or you have purified the protein and then you have sequenced. No, you got from the nucleotide, but still it will be considered a primary database. Okay, because this has come from the experimental outcome. Similar way, there is similar way there is several database which holds the protein sequence which consider under the primary database. So very common is the protein information resource, protein sequence database. So this is a collaborative endeavor between the PIR, Munich Information Center for Protein Sequence Germany, and the JPID. JPID is the Japan International Protein Information Database. So this is a collaborative between these three. Okay. The PIR PhD is now a comprehensive, non-redundant, and expertly annotated. Annotated means each and every informations are available. Like there is two types of data. One is the raw data, another is the annotated data. So they are having very nice annotation. Okay. A unique characteristic of the PIR PhD is its classification of the protein sequences based on the superfamily concept. The sequence in the PI-PRSD is also classified based on the homology domains and sequence motifs. There is another database like PIR PhD is the SwissProt. So this is also this curated protein sequence database also provides a high level of annotation. This is also equivalent to good actually like a PIR PhD. There is some other database actually that called the PDB. Okay, just now when Dr. Khan was introducing me, actually he has told about the coordinates, structural coordinates in the protein data bank. So this is the protein data bank. In the protein data bank, you don't submit only the protein sequence. You submit protein sequence along with the structural information. So this structural information, from where you get actually structural information. Protein sequence, yes, you can get from the SwissProt. You can got from the GenBank after the translating the nucleotide sequence. But from where you will get the information for the structural coordinates or protein structure. So for that you have to determine the structure of a protein. So when you want to determine a structure of protein, anyway in the coming classes I am going to tell you much about that. So basically there is a three experimental methods, because here these all primary data is for the experimental data. So you can determine experimentally by three methods. One is the X-ray crystallography, another is nuclear magnetic resonance (NMR), and third one is the cryo-EM, that also called the cryo-electron microscopy. So all these three methods give you the three-dimensional structure of a protein. So this database, which called Protein Data Bank (PDB), is having information for the protein structure and protein sequences all together. Now you will say only this having the protein data, protein information. No, this is having the information for the nucleic acid also, like RNA, DNA. There is information for some other biological molecules, like even some carbohydrates structures are also available, because some carbohydrates binds with the protein. So then you can determine structure with that. There is some protein which is nucleoprotein, so nucleoprotein structure is available. So then they are available along with the nucleic acid. So all those structures are available, and even structures of the proteins are available with the different ligands. Different ligands like a substrate, like a cofactor, such as ATP, like antibiotics, like gramicidin. So all these things are available in the database. So you can collect this information from this database and use for your scientific purpose. There is another database, like I just mentioned, for like a nucleotide. We are having two types database. One is the primary, another is secondary. Similar way, protein is also having the secondary database. So primary database, when you are having the protein sequence there. Secondary database, people have derived the information from this primary database, and then they have submitted in a database. That database will call the secondary protein database. So one example is the ProSite. So ProSite is having the motif and pattern of the protein, whatever proteins is available in the primary database. Then another data is the database called the secondary database is the prints, which is having the fingerprint of the protein sequences. And another is the MHC PEP. So you know MHC is the major stability complex. So this is having the peptide sequence which bind to the MSC, major histocompatibility, major histocompatibility complex of the immune system. And another database is the P form. As you see by the name, P means protein, form means family. So in this, they have defined different families on the basis of the four elements. One is the annotation, second is seed element, seed alignment, third one is HMM profile, and fourth is the complete alignment. So on this basis, they have defined this P form. So these are the just basics about the database. So now if you want to just differentiate what is the difference between the primary database and the secondary database. So primary database is the archival database, like experimental database, and secondary is the curated database or knowledge database from the primary database. Source of the data is the direct submission, because whatever experiment you have performed, you are submitting in the primary database. And for the secondary database, source is the whatever is submitted in the primary database. From there, you have done analysis. You have retrieved information on the basis of literature, research, and interpretation. That is the part of the secondary database. These are the few examples which I have just told you in the previous slides. Uh, so now I will bring you the experimental part. This is just some basics of the database. So in the database, because I will mention you there are several databases, primary, secondary, for protein separately, DNA separately. So what way you have to get the information from there if you want to perform a sequence analysis. If you want to perform a sequence analysis, you have to get information in a FASTA format. So the question comes, what is the FASTA format? FASTA format is a text-based format for representing either nucleotide sequence or the peptide sequence in which base pair or amino acids are represented using single letter codes. The sequence in FASTA format begins with a single line description. As you can see here in the bottom, the description. In this box, I have given one example how it looks actually. I'm pretty sure mostly people are familiar, okay, with these things. If you are not, please you can stop me and ask further actually. And anyway, when I am going to show you, um, uh, when I am going to practicing you, you can ask more questions. So with a single line description, so this kind of description you can see. In this kind of description, you are getting several words actually. Okay, this HM2359117.1. So this is basically the accession number. Then what is this? So this is a Sisser aratinum cultivar Pusa 256 lectin gene, and it is having the complete coding DNA sequences. So a sequence in FASTA format begins with a single line description followed by lines of the sequence data. The description line, the description line is basically will start actually with this symbol which we call greater than signal, greater than symbol, and then it is recommended that all lines of the text be shorter than the 80 characters in the length. So this is one example. Now, before moving further, I just want to take you how to access the uh, as primary database. So I just want to give you one example. If possible, please you can also move along with me, and I will share this my screen. Are you able to see my screen?
स्क्रीन दिखाई दे रहा है लेकिन कुछ ग्रीन नजर आ रहा है बीच में.
Okay, okay. So let me see.
अब गया, ठीक है.
Is it okay now?
It is fine, sir. Now.
Now you can see this Firefox. So, like now I need your input also. Please join me. Actually, there is not any problem. Actually, this is scientific discussion. So, please try to be. Please try to follow me. Actually, whatever I am doing, you can try parallelly. And if there is any doubt, you can ask me. Okay. So let's now in the because just now I mentioned you there is 3 primary database. Okay. So we will go firstly for the GenBank. So let's put in the your search place, put GenBank. Are you able to see this thing?
Sir?
Dr. Khan, is it possible to talk to these people actually?
Participants, I have logged the, when to say, unmute, I will make them unmute.
Yeah, please unmute them actually. So, because this is more interactive actually. So, if they will tell me yes, whether they are able to follow me or not.
Sir, I have made unmute to them. They can speak.
Yes. So, are you able to follow me?
Yes, sir.
Yeah, thank you.
Yes, sir. Yes, sir.
Yeah, so let's go to Google, and in the Google just put one word GenBank. And please follow this screen. Whatever I am doing, you please follow this thing. Instructions also. Okay, so after reaching this, click on the GenBank. Okay, so now you're able to see this screen actually. So in this screen, like because in beginning I told you primary database is made for to get the nucleotide sequence information. Even you can get the protein sequence also. But we want to start with the nucleotide because in the lab, if you are going to determine a sequence, that will be the nucleotide sequence. It is difficult to get the whole protein sequence easily. But nucleotide sequence just 250 rupees or 300 rupees you give to any company and they will give you the easily they will sequence your primer plasmid. Okay, so go here and just put one code AY221982. Okay, after putting this code, start search. Okay, I hope all of you have reached here. So here you can see clearly because this one was one code actually that code called accession number. So because this is my own accession number, so I remember the number. But if you don't know the name of the particular protein or particular gene and you want to look, like suppose you want to look at chickpea lectin. Just put here information and here you will get lot of proteins, okay, which is sorry, lot of nucleotide sequence which is submitted in the GenBank as a primary sequence. Okay, now here you can click and then you can reach to the same sequence which I was showing you. That is the AY221982. So I hope all of you are able to follow me and you have reached here. Okay, so now just I will show you what is this sequence actually. What information you have to retrieve from this sequence. So first thing, what is the locus? Okay, locus is the AY221982. What is the length of this sequence? So this length of this sequence is the 972 base pair, and this is a basically mRNA. Why it is mRNA? In the bottom, I will explain you why they are calling it mRNA. This is a linear sequence because this is not a double strand, so we will call this a linear sequence. Okay, this was submitted long back actually when I was doing my PhD. Okay, and here you will get see the all details. What is the accession number? What is the definition of this sequence? Means what is the description of this sequence? So this sequence actually is a lectin mRNA which is derived from the Caesar-Aratinum. Caesar-Aratinum is the scientific name of the chickpea. Okay, so then all those things are there. Source, Caesar-Aratinum chickpea. Okay, the organism is chickpea, that is basically we know it is eukaryotes. So all that classification is here. Then references and then who is the author. So as you can see here, I am the author, Qureshi IE, and this work was published long back in the 2007 from my PhD in the 2007. Okay, so now here you come in the bottom. In the bottom you can see 12972 amino acids. Now these amino acids are available as a nucleotide sequence. These nucleotide sequence are available as a simple mode, but these numbers are coming here. These numbers will interfere when you want to analyze of this sequence. So that is the reason we need FASTA form of this sequence. So you click on the FASTA format. Now when you will click on the FASTA format, you will reach on the sequence. So now you are able to see sequence. There is not any kind number. If you remember, I am telling this is mRNA. Why this is mRNA? As you see at the end of the sequence, because this sequence I have isolated from a cDNA library. So if you will isolate from the cDNA library, what will be the difference between the cDNA clone and genomic DNA clone? Genomic DNA clone will not have the poly A tail, but cDNA clone may have the poly A tail, and you can see at the C terminus, at the 3' of this sequence, you are able to see the poly A tail. Okay, so that is the reason their code they are asking this is a mRNA, okay, because it is containing a poly A tail. Because this one we have derived from the cDNA, and cDNA comes from the mRNA. So now we got the FASTA format. So you see, as I told you, in the FASTA format, generally it starts with a description. So description is this one, AY221982, and it starts with a greater than symbol, then AY221982, and point 2 means which format this one, which version this one. So in the beginning, I have submitted a truncated version, and later I have updated. So when I update it, then this one will call the second version, not first version. If you see just now in the slides, that one was the first version, but this is the second version. So then they are saying point 2. Okay, Caesar-Aratinum, the organism name, lectin, the name of the gene, and because it is derived from the cDNA, so they are saying mRNA, and partial cDNA. Actually, when I submitted first time, it was not having the 5', like 5' was missing actually. So then it was that time partial CDS. So they are considering from that time only. Later I have extended, and now you can see it is starting with the ATG. So it is having now complete CDS coding DNA sequences, but because once they annotate, then they cannot change annotation. So the annotation will be same, only version will change. So version has changed, but description is the same. So that is the reason. So that is not a big problem actually. So but this is having the full length of the protein, full length of the gene. Now if I take you back actually, that is the FASTA format. Now you will ask me how to get a protein from this FASTA format. So what you can do, first simple thing actually, you can go to the Google and say one word, because if you see in this sequence, there is a several sequence which is not required for the protein. part actually. That is only part of the because upstream you cannot see because it is starting with the ATP. But you can see in downstream lot of is there, especially this polyethylene. So how to get a ORF from here? Because you are having now gene with you and you want to get a ORF. ORF is the open reading frame. From there you can express a protein which translate in a protein. So now let's go to the another tab and put a word ORF finder. Are you able to reach there?
Yes, sir.
Yeah, thank you. So now after reaching here, you click on the ORF finder. So this ORF finder, what is the ORF finder? Let's firstly, let me tell you about something about the ORF finder. So RF finder identify the all open reading frame, ORF finder, reading frame or the possible protein coding region in sequence. It shows 6 horizontal bars corresponding to one of the possible reading frame. In each direction of the DNA, there would be 3 possible reading frames. So total, you will get the 6 reading frames, and they will be tagged like a +1, +2, and +3. And reverse in the reverse system, they will consider -1, -2, and -3. The resultant amino acids can be saved and searched against the various protein databases using BLAST for finding similar sequence or amino acids sequence. So this is the ORF finder. So in the ORF finder, you are having full freedom to either you get the accession number. So as you remember, we know the accession number of this sequence, that is the AY221982. But in some cases, what happens? You got a new sequence, new gene, you have sequenced that, and now you got the sequence only because you have not submitted in the database. Just you want to see whether this sequence is having homology with your whatever you are amplifying or it is a novel gene. So for that, we generally take the nucleotide sequence. So let's go to the again FASTA format and copy this sequence and paste here. And after pasting, now you go down actually. When you will go down, you will get some parameters like a what is the minimal ORF length you are looking. So you are looking at ORF length, it's just 75, or it is a 30, or it is a 150, or it is a 300, or it is 600. If you see the length of this gene is 972 base pair. So you can assume that definitely ORF will be bigger than to the 600. Still, if you are working with a small protein or a small gene, then you can go for the small ORF also. Now next question, it will ask you genetic code. Which genetic code you want to follow? So because this is from the Sisal Aritinum, so we are assuming that this will follow a standard because we are not talking about the yeast, we are not talking about the mitochondria and specific things. Only we are talking about the standard one. So let's go for standard one. Then the next question it will ask you, next parameter in this server is the start codon. What can be the start codon? So if you see your whole sequence, whatever you have pasted, in this you can see in the beginning itself ATG. So that's mean yes, ATG only. But in some cases, if you are not able to see the ATG or you don't want to take any risk, just click any sense codon or ATG and alternative initiation codon. Because if suppose in your sequence you are not having ATG and you have clicked on the ATG, then it will not able to give you the ORF. So then it is better to click in that sense ATG and alternative initiation codon or this any sense. But here because we are aware, so we are keeping ATG only. Okay, then now let's submit the job. When you will submit the job, it will perform its analysis, and then at the end it will give you the different open reading frames. And just now I mentioned you will get open reading frame in that both direction, plus direction and negative direction, because it will reverse your sequence and then it will give you. As you see here, it is giving you almost how many open reading frames? If you see, it is giving you almost 14, 15 open reading frames. Why? Because you have taken very small number of open reading frames. So you can see here, even it is giving you the open reading frame just having the 32. So like let's come to this table actually. I hope all of you are able to follow me. So ORF1. So ORF1 is coming from the, because DNA is having two strands. One is the positive strand, another is the negative strand. So because you have to look firstly in the positive strand. So ORF1 is coming from the positive strand. So it is belongs from the first frame only. Okay, so it is starting from the first nucleotide. Because if you remember, first nucleotide in your sequence is the ATG. Then it is having the stop codon at the 807 nucleotide. Okay, that is the reason the length of your protein or your protein will contain around 268 amino acids. If you convert 807 nucleotide in the amino acids, how many amino acids you will get? 268 or more or less?
268 only.
If you will, if you just you divide 807 by the 3, how many you are getting?
269, sir.
Yes. Why 269 is there?
One stop codon.
Yes. Good. So because it will tell you only the ORF region. The stop codon will not be part of ORF region. So then it is showing you only the 268 amino acids. But yes, it is saying ORF region is the 807 nucleotide or base pair. So this way it will give you the several reading frames. Okay, ORF9 which has come from the negative strands, means reverse strand. Okay, that is starting from the 477 to 211, but the length is very small, just 88 amino acids. So this is itself is indicating you, yes, you are having something quite bigger. And now you see the same thing here you are having as a picture. Okay, so this one is the top one. Same whatever is present in the table, similar thing is present above as a picture. So you can see this clearly what is there actually, from where it is starting and where it is ending. So all these information you can get. And now because you are getting lot of frames, what you can do, you can reduce the numbers actually. What you do, you go modify your research, modify your search and reduce the number, because I have taken the highest number. That is the lowest number, that is the 30. Now what we can do, we can reduce this same number to the 600.
ఆల్ ద బెస్ట్. తేజు మీరు కూడా ఐ మీన్ మీరు కూడా ఎక్కడన్నా ఇంటర్మీడియట్ ఎక్కడో చోట ఇనిషియల్ గా జాయిన్ అయిపోతే అప్పుడు తెలుస్తా ఉంటది మీకు, దెన్ యూ కెన్ మూవ్.
So now you see, for the
ఉండడానికి ట్రై చేయండి. 1 year 97. ఓకే,
please mute yourself, madam.
వస్తే కానీ అది పర్సెంటేజ్ దాంట్లో పోతా ఉంటది. ఏం పర్లేదు, ఇప్పుడే కదా, స్టార్టింగే కదా.
Please mute yourself.
I think 1 or 2 they kept cut.
Okay. Yeah, so now you see what I did, actually I just changed little bit parameters. Instead of having 30 base pairs, now I changed to the 800. Now when, sorry, 600. When you see, when you change the parameters, now you are getting only the one open reading frame. Why? Because other frames are very small actually. The length was very small. So now you are not getting any negative, because negative, if you remember from negative strand, that was around 200 base pair. So that will also not fit here, because we are having the threshold 600. So this way you can play and pick up a ORF for your gene sequence. Now, after getting ORF, what should we do actually? What is the next after doing ORF? So now you got a protein sequence from a gene. Now you want to do further analysis. So what could be the further analysis? Can we directly take this one and clone the gene? Because this has been translated from a gene. So can we take those 807 amino acids, 700 base pair, and we can directly do, or we have to recheck further. So before going that, I want to tell you one more thing actually about the, now when you get any sequence, like suppose I got this sequence, I should firstly check whether this sequence is having any homology with the available database or not, because that will give you a lot of information. Like suppose this is already annotated now, but if this was not annotated and you have sequenced by your own and you got a sequence, primary sequence of nucleotide, and now you want to know what you have to do. So for that, generally people go for the alignment. So alignment is also having two types. One is the global alignment and another is the local alignment. So anybody can tell me the difference between the global alignment and local alignment? See, we all are teacher actually. So and teachers should be very intellectual actually, interactive. So okay, so nobody wants to answer, not a problem. Okay, so there is two types of the alignment. One is the local and another is the global. So just I will show you one example of global and local alignment. Okay, you see, you are having one sequence here. Now this sequence when you will put in a data, when you will put for the alignment, you can see the comparison for the whole sequence. Means this will look for a template and then it will this will set a template and then it will align with it. Okay, compare with that. So now you see, even there is a gap, it won't bother. It will give you the sequence alignment. Okay, that this much region is aligning while this much region is not aligning. That is the basically global alignment. Local alignment, what happens in the local alignment? Local alignment will tell you only the region which is having the homology with the template. Okay, so a small seed is uncovered that can be used to quickly extend the alignment, and now the extended alignment. So this way you see now, it is not telling you about this region or this region. It is telling you only this region which is having homology with the template. So this is the difference between the global and local alignment. So when we talk about the global and local alignment, so let's firstly look at global alignment for our protein, and then we will go for the firstly we'll go for the global alignment and then we'll go for the local alignment. And before that, what you can do actually, you can blast this one. So there is one software server actually that's called blast. So because we are having the nucleotide sequence, so we can put blast in. Okay, so blast n is for the nucleotide blast. Blast P is for the protein blast. Okay, so just click here, and after clicking, then this will open, and then you go and take your sequence. Okay, copy and paste your sequence here. I hope all of you are able to follow me. Okay, if any doubt is there, please stop me so I can help you to solve your problem. Okay, so now when you will put your query here and you will check in the database, there is several database actually, like a RNA ITS database, genomic transcript database, beta coronavirus database. This is new database actually, it has come due to the ongoing COVID-19 problem, and another is the standard database. Generally, we prefer standard database, and because we don't know information what is there actually, so we will go simple for the nucleotide collection. Yes, if you are sure, yes, which organism this is. Like suppose we know this is from the chickpea. we can check in the chickpea genome. Okay, or if it is from human, okay, then you can put here human and then you can search against that. So we will not do that thing. So here you can include or you can exclude also. Like suppose, because we know this is from chickpea and I don't want to compare with the chickpea. I want to compare with the other leguminaceae family member like a pigeon pea, okay, like moon, other members of the this family. So then I can write here chickpea and then I can exclude. So then what will happen? It will not consider chickpea. Okay. Then these are the different parameters, and after that you go for the highly similar sequences. So always first attempt with the highly similar sequences, because if you will pick up more dissimilar sequences or somewhat similar sequences, then lot of template you will find actually. And you want to perform the now alignment. So for alignment, you should firstly know who is having the highest homology with your sequence, nucleotide sequence. So then most preferred one is the highly similar sequence. There is other parameters also like you can change the target sequence length, you can change the threshold and other things. But these are let it be for time being like same and don't change other parameters. Let it be default and then click the blast. Because now what it is doing actually, it is looking in the database, whatever the database is available. So it may take some time actually. Okay, so yes, but it is very prompt actually today. So we have to be happy. Okay, so now it has given the result. So if you see here, the program you have performed blast N. You cannot run blast P actually for the this because this is a nucleotide sequence. Okay, and what is the query length? Because if you remember here, we have checked in the beginning itself, it is 972 base pair. So then it is showing 972 molecular type. What is this? Because it is N, so it can be RNA molecule also. But yes, it is a DNA molecule. Okay, now you see, it is giving you the whatever is the best fits are possible. It is giving those things. Okay, like what is the maximum score, what is the total score, what is the query coverage. Query coverage is very important factor actually, and this is I will describe in the coming classes more actually when you are determining a structure. That time query coverage is very important. Now if you see, because this one query coverage is 100%. Why it is 100%? Because you have picked up same sequence in the database AY22198. But if you see with the other one which is available, okay, now if you compare with this, you can see there is some changes actually, because this is isolated from which plant? This is same lectin gene is planted is isolated from the P sativum. P sativum is the Plasmodia, sorry, Pisum sativum, that called also green pea. So this is derived from there. The length of the is the 953 base pair, while your length is the 973. If you see the homology, it is there is two characters. One is the identity and another is the similarity. Anybody can tell me the difference between the identity and similarity? See, I'm just asking this question actually. So if you are not aware, then definitely it is my duty to tell you. Okay, so what is the identity and what is the similarity? Okay, because in the case of the nucleotide sequence, you cannot say the similarity. Only you can talk about the identity. Like if you are having A means only A. If you are having A, then it is identical. If you are having C here, then it will not consider identical and it cannot be similar also. But if you see, because in the column with the nucleotide, because you are having only the 4 nucleotide base. But if you see in the case of the protein, there is 20 amino acids. So 20 amino acids are having identity also and they are having similarity also. So that is the reason in this case you are able to see only the identity. So here you are able to see only the identity and that is showing it is 98% identity. So by this you got the information there is a sequence which is having the high homology with your protein. So now because you want to do the alignment, For alignment purpose, it is good actually that you are having that information, and on that basis now you can proceed further. Okay, so you see here they have given you the accession number also AY221982, as we know because it's we have taken. Same thing X66368.1, because this is that point 1 and point 2 depends on the version, which version this is. Version 1, version 2, version 3. So if you see here in this table, because you are having a gene from the chickpea and you see it is having homology with the Pisces-cation, that is another leguminous member. Then you see Cagnus cagen, that is the pigeon tree. Okay, this Cagnus cagen is basically a hair dal or pure dal. Okay, so all these are having lens nigricans. This is a move actually. Okay, so this way it is having the all beginner data. All these things are there. Okay, so if you see here, all leguminous members are there because generally, if any protein you take, they are having high homology within the family. So it is indicating you, see, it is having 100% query coverage, it is having 98% query coverage. If you see identity, it is having 100% identity because it is same protein. But if you see with the other proteins, also it is having 98%, 98.32%, 98.11%, and other things. Even using this one, you can make a phylogenetic tree also, whether they belongs from the same family or not. Okay, so you can pick up sequence here, and then you can go and just have a. This is a graphical summary. Okay, now you see here, the red one means it is having 100% homology. Okay, very high homology. And if you see in the some sequences, you can see some gaps are there. Because for this reason, either they are not having the sequence or sequence information is not available. So then they are having this gaps. In this reason, you see this region is missing because this sequence is not present in the sequence which they are having in the database. While this area is present in your sequence, but in other sequence. So now let's come back actually on the sequence alignment. So as I mentioned, there is two types of the sequence alignment. One is the global and another is the local. So let me show you one example of the local alignment. So align blast, just please type in the Google and let's see. What is there? Okay. So now it is telling you, it is giving you several options, like which I told you in beginning, like you can do nucleotide blast with the blast N. Okay, you can translate the nucleotide sequence in a protein and then you perform, and then you can do the blast N also. Sorry, okay. Okay, so I hope all of you are able to follow this thing. So here you can see the two boxes. One is for the whatever query you are having and whatever you are having the subject. Query means, like suppose we got the sequence from here. So this is our query. Okay, put that sequence here. Then subject, because if you remember when we were looking for the chickpea lectin, there is another group actually who has isolated. Yes.
आपका स्क्रीन मूव नहीं हो रहा है.
But it is moving.
हाँ.
Okay. So is it there now?
हाँ, अब दिखाई दे रहा है.
Yes. So there is two columns you can see clearly here. One is the query sequence and another is the subject sequence. So because if you remember, like just now when we were looking for the chickpea lectin, there is another sequence of the chickpea lectin. Actually, this is also isolated from the same lab from where I have performed, from where I have done my PhD. Okay, so what we can do, we can take the faster sequence of this to know what is the homology between these two sequences. Okay, so now this sequence has come. This is a little bit smaller sequence than that sequence, but let's copy this sequence also and go to the subject of the align and paste there. Now in this again same parameters will come whether you want to look for the highly similar sequences region or more dissimilar sequences or somewhat similar sequences because they are coming from the same plant actually, chickpea. So what we'll do, we'll look for the highly similar sequences. So let's blast this one. Okay, and then wait for the some time. Meantime, what we'll do, we will go for the parallelly because we are having a sequence. Okay, now you see here, it has performed this alignment. And now in this alignment, you will see description. So because we have not given any heading, so it is showing none provided. Then another is the graphical summary. Okay, if you see, just now I mentioned red means you can see this legend on the top actually alignment score. If you are having less than 40% identity, then you will get black line here. If you are having 40 to 50% homology, then you will get a blue line here. If you are having 50 to 80% homology, then you are going to get the green color, and if you are having 80 to 200%, then it is going to give you the pink color. And if you are having more than 200%, okay, your score 200, more than 200, then you will have the red line. So you see this red line is indicating that yes, they are highly homologous. So now let's go for the next thing is the alignment. Now let's align this one. So you can see here on the top, the score is very high, 662. Then yes.
Sir, lunch break लेके हम continue करेंगे ना, please.
Okay, yeah, no problem, sir. Just let me finish this one, sir. Is it okay?
जी, lunch break के बाद continue करेंगे क्योंकि 12:40 हो गया. वो 1:30 को फिर participants को join करना है.
Oh, okay, sir, no problem.
Sorry sir, आप मतलब अच्छा ले जा रहे थे topic, लेकिन अभी वो lunch का time चालू हो गया. 12:30.
Yeah, I'm sorry. I didn't check actually time. It is my mistake.
मैं माफी चाहता हूँ. Actually lunch break चालू हो गया तो participants धीरे-धीरे निकलना शुरू हो गए. तो lunch break दे देते हैं और उसके बाद फिर join कर लेते हैं.
Okay, sir, no problem, sir.
फिर meeting चालू कर देंगे.
Yes, sir. Okay, 1:30 we will start.
Okay, I beg pardon. But dear
Sir, what time will start, sir?
जी.
What time will start, sir?
1:30.
1:30. Okay, sir.
जी.
Okay, thank you.
Yeah. Dear participants, please join back at 1:30 because you are now going to have the hands-on of the in silico sequence analysis. As sir has already started, but just because of lunch break, I have requested him to stop here. Please join back again sharp at 1:30 so that we can have more insights on how we can go for the in silico sequence analysis. Thank you, and with permission of sir, I want to end the meeting now, and we'll be back again at 1:30 sharply.